1. Unlocking autism's complexity, for biologically distinct subtypes discovered. 2. For autism subtypes linked to unique genes and traits, new study explained. 3. Unveiling 4 new autism subtypes linked to genetic variations. 1. Unlocking autism's complexity, for biologically distinct subtypes discovered. What if we told you autism isn't one condition, but 4 distinct biological subtypes? A groundbreaking study led by Princeton University and the Simons Foundation just reshaped our understanding of autism's genetic roots, offering a roadmap for precision diagnosis and care. But here's where it gets controversial. This discovery challenges decades of assumptions about how we categorize and treat autism. Let's dive into the details and explore why this could spark heated debates in the scientific and clinical communities. For years, autism research has grappled with its complexity. Autism isn't a single disorder but a spectrum of conditions with overlapping traits. Now, researchers have used advanced computational models to analyze data from over 5,000 children in the InSpark study. https://sparkforautism.org/portal/page/about-spark, funded by the Simons Foundation. Instead of focusing on isolated traits like repetitive behaviors or social challenges, they took a person-centered approach, examining over 230 traits per individual. This holistic method revealed 4 biologically distinct autism subtypes, each with unique genetic patterns and developmental paths. Their findings, published in Nature Genetics, https://www.nature.com/articles/S41588-025-02224-C, could revolutionize how we diagnose and support individuals on the spectrum. Let's break down the 4 subtypes and what they mean. 1. Social and behavioral challenges. This group, representing 37% of participants, displays core autism traits like social difficulties and repetitive behaviors but typically reaches developmental milestones, walking, talking, on par with neurotypical peers. Many also face co-occurring conditions like ADHD or anxiety. Think of it as a subtype where autism coexists with other psychiatric challenges, but motor and language skills develop at a typical pace. 2. Mixed ASD with developmental delay. Around 19% of participants fall into this category. These individuals often experience delays in motor and language skills but show fewer psychiatric symptoms like anxiety or disruptive behaviors. The term mixed reflects variability in social challenges and repetitive behaviors within the group. It's a subtype where developmental delays are prominent, yet emotional regulation is relatively stable. 3. Moderate challenges. Covering 34% of the study population, this group exhibits milder autism traits and follows a developmental trajectory similar to neurotypical peers. They rarely face co-occurring psychiatric conditions, making it a more isolated form of autism. Picture a person with autism who may struggle socially but doesn't require intensive support for other mental health issues. 4. Broadly affected, the smallest group, 10% of participants, these individuals face severe, wide-ranging challenges. They often have developmental delays, communication difficulties, and co-occurring conditions like mood dysregulation. This subtype is the most genetically complex, with the highest rate of damaging de novo mutations, genetic changes not inherited from parents. But here's the twist, these subtypes aren't just clinical labels. They're tied to specific genetic mutations and biological pathways. For example, the broadly affected group has a higher prevalence of de novo mutations, while the mixed ASD group often carries rare inherited variants. This distinction matters because it suggests different underlying mechanisms for seemingly similar symptoms. As co-lead author Avia Littman explained, it's like solving multiple jigsaw puzzles at once. Without separating them, we couldn't see the full picture. And this is the part most people miss. The timing of genetic disruptions also varies. In the social and behavioral challenges group, mutations affect genes that become active later in childhood, explaining why these individuals often receive a later diagnosis. This challenges the long-held belief that autism's genetic impact occurs prenatally. As researcher Chandra Thiesfeld noted, we're mapping autism's trajectory from biology to clinical presentation in ways we never could before. So, what does this mean for families and clinicians? precision medicine is no longer a distant dream. Knowing a child's subtype could guide tailored interventions, from targeted therapies to school accommodations. For example, a child in the broadly affected group might benefit from early intensive support, while someone in the moderate challenges group might thrive with social skills training. But does this mean we're oversimplifying a complex condition? Critics might argue that rigid categories risk missing individual nuances. The study's implications extend beyond autism. Its framework could help classify other complex disorders, from cancer to neurodegenerative diseases. As co-author Natalie Sauerwald said, this opens the door to countless discoveries. Yet questions remain: will these subtypes hold up in larger, more diverse populations? Could they lead to new treatments or even preventative strategies? What do you think about this new approach to understanding autism? Does categorizing it into subtypes empower families, or could it limit our view of individual differences? Share your thoughts below. Let's keep the conversation going. 2. For autism, subtypes linked to unique genes and traits, new study explained. Researchers identify 4 distinct autism types with unique genetic signatures. The traditional view of autism as a single condition with varying severity is being challenged by groundbreaking research. A fascinating new study published on July 9 in Nature Genetics reveals that autism may actually comprise 4 distinct subtypes, each with its own genetic fingerprint and characteristic traits. This discovery could revolutionize how we understand, diagnose, and support people with autism. Michael Lombardo, senior researcher at Italy's Istituto Italiano di Tecnologia who wasn't involved in the research, confirms this supports the emerging concept of many autisms, rather than a single spectrum. But here's where it gets interesting: identifying these genetic connections is merely the first step. The real challenge now, Lombardo emphasizes, is unraveling the biology that stems from those genes. The research team, headed by Princeton University's Professor Olga Troyanskaya, who also serves as deputy director for genomics at the Flatiron Institute, employed sophisticated computational modeling to analyze an extensive dataset. They examined information from 5,392 autistic children between ages 4 to 18 and their non-autistic siblings collected through the SPARK study. This comprehensive data encompassed genetic findings alongside parent-reported information about developmental milestones, cognitive and behavioral characteristics, co-occurring conditions, and family medical history. Using advanced machine learning techniques, the researchers identified 4 distinct autism subtypes based on both genetic markers and clinical presentations. The social/behavioral group constitutes the largest segment at 37% of participants. These individuals display repetitive behaviors and communication difficulties alongside various co-occurring conditions, but show few developmental delays. Their genetic profile includes common variants linked to ADHD and depression, plus rare variants in genes that become active primarily after birth in specific brain cells. The mixed ASD with developmental delay group represents 19% of the sample. These children reach developmental milestones significantly later than neurotypical peers but typically don't experience co-occurring conditions like anxiety or depression. Their genetic signature includes numerous rare, potentially harmful variants, both inherited and spontaneous, in genes active during early brain formation in the womb. The moderate challenges group accounts for 34% of participants. Their developmental trajectory resembles the social/behavioral group but with less pronounced symptoms. Genetically, they carry rare alterations in less critical genes, potentially explaining their milder core autism characteristics. The smallest segment, the broadly affected group, 10%, exhibits prominent autism traits from early childhood. These individuals carry a substantial burden of rare mutations in crucial genes, including targets of a protein implicated in Fragile X syndrome. And this is the part most people miss: these genetic differences correlate meaningfully with developmental timelines and diagnosis age. For instance, groups with variants in early expressed genes, the broadly affected and mixed ASD with developmental delay groups, demonstrated delays in early developmental skills and received earlier diagnoses. Conversely, the social/behavioral group, with variants in genes expressed after birth, received later diagnoses and follow developmental trajectories more similar to non-autistic children. There are a lot of kids who seem very neurotypical until a bit later in childhood, explains study investigator Natalie Sauerwald, associate research scientist at the Flatiron Institute. The fact that we were able to find genetics that aligns with that was really surprising. While previous research has attempted to identify autism subgroups, no definitive classification system has emerged, likely because results vary depending on study populations and methodologies. Lombardo points out that this new study's broad age range, 4 to 18 years, increased sample size but introduced variability since children at different developmental stages were grouped together. His own research, which focused on narrower age ranges, identified fewer subtypes, suggesting that different datasets may yield different classification systems. What sets this study apart is its holistic approach. Rather than connecting genes to isolated traits as previous studies have done, Troyanskaya's team examined each person's complete constellation of characteristics. To validate their findings, the researchers cross-referenced their results with data from the Simons Simplex Collection, SSC, which contains clinician-gathered information. The consistency between parent-reported data and clinical observations strongly suggests these subtypes reflect genuine differences, according to Troyanskaya. As additional data becomes available, these autism subtypes may be further refined, potentially revealing more detailed genetic and clinical nuances within each group. Expanded datasets could validate these findings across more diverse populations and adult cases, potentially leading to more personalized diagnoses and support strategies in the future, Sauerwald suggests. Thomas Bergeron, director of the Human Genetics and Cognitive Functions Unit at Paris's Institut Pasteur, who wasn't involved in the study, agrees with this assessment. Some individuals need medical support, maybe gene therapy, and others need better inclusion in society, better recognition of neurodiversity and so on, he explains. However, Bergeron highlights a common limitation in autism research, an overemphasis on clinical data at the expense of real-world experiences. He advocates for more practical, longitudinal information to better understand autism's impact on daily functioning. We need to have a better idea of the trajectory of these individuals. Do you think these distinct autism subtypes will lead to more personalized support approaches? Or could categorizing people potentially limit our understanding of autism's complexity? Share your thoughts in the comments below. 3. Unveiling 4 new autism subtypes linked to genetic variations. Unveiling the autism spectrum, a new perspective. The autism spectrum, a diverse landscape, has just become a little clearer. A recent study has identified 4 distinct subtypes, each with its unique genetic signature and set of challenges. July 10, 2025. 5-minute read. Researchers uncover 4 autism subtypes, linking genes to traits. Imagine a spectrum as vast and varied as the night sky, each star representing a unique experience. This is the essence of autism, a condition with an incredible range of presentations. And now, a groundbreaking study has shed light on this diversity. The autism spectrum, a complex landscape. Autism or autism spectrum disorder, ASD, is a complex condition, and as Stephen Shore, an autistic professor, famously said, "If you've met one person with autism, you've met one person with autism." This quote resonates deeply with researchers and the autism community, highlighting the unique challenges and experiences that fall under this diagnosis. Fred Volkmer, a psychiatrist and professor emeritus at Yale University, adds, "The beauty of the autism spectrum lies in its heterogeneity, but it also hides the differences." Currently, individuals are placed on this spectrum based on the severity of social communication difficulties and restricted, repetitive behaviors, categorized from level 1 to level 3. However, these broad categories often fail to capture the intricate nuances of each individual's experience. This is why researchers have been tirelessly working to divide the spectrum into meaningful subtypes, using genetics and behavioral characteristics as their tools. A breakthrough study. In a study published in Nature Genetics, researchers have made a significant breakthrough. By analyzing data from over 5,000 autistic children, they identified 4 distinct subtypes of autism, each with its own set of challenges and linked to specific genetic variations. The 4 subtypes. 1. Social and behavioral challenges. This group, comprising 37% of participants, faced more difficulties with social communication and restricted behaviors than other autistic children. They also had challenges with disruptive behavior, attention, and anxiety, but without significant developmental delays. 2. Mixed ASD with developmental delay. These children, 19% of the participants, showed a varied range of social communication and restrictive behaviors, along with some developmental delays compared to non-autistic children. 3. Moderate challenges. This group, 34% of participants, had fewer difficulties with social communication, restrictive behaviors, and other core autistic traits compared to other autistic children, but still more so than non-autistic children. They did not experience developmental delays. 4. Broadly affected. These children, 10% of the participants, faced severe and wide-ranging difficulties with social communication, restrictive behaviors, and other core autistic traits, including developmental delays, compared to other autistic children. While these groups still contain variation, participants within each group share more similarities with each other than with those in other groups. The study's findings were also replicated in a smaller population of autistic children. The genetic connection. What's remarkable is the distinct genetic profiles of these four groups. Olga Troyanskaya, a genomics researcher at Princeton and the study's senior author, says, "The biggest surprise was how different the four subtypes turned out to be. The underlying genetics and biology are very different. For instance, different genes become active at different developmental stages, some before birth and others post-birth. The social and behavioral challenges group had more mutations in genes active post-birth, which could explain their lack of developmental delays and later diagnosis compared to the other groups." Implications and future directions. Catherine Lord, a psychologist specializing in autism at the University of California, Los Angeles, notes, "The groups make sense and follow lots of findings by other researchers." Fred Volkmer adds, "It's like rediscovering the wheel, but the connection to genetics makes these results most noteworthy." However, it's important to note that these four subtypes may not cover the entire autism spectrum. The study's sample, largely white and with European ancestry, may not represent the diverse autistic population. Additionally, some rare traits related to autism might not have been captured due to limitations in the available data. As Natalie Sauerwald, co-lead author of the paper, says, "Understanding a child's specific subtype can provide greater clarity and open doors to personalized care and support." This study highlights the need for a more nuanced approach to autism diagnosis and understanding. As Volkmer puts it, it spurs us to think about new approaches, with subtypes rather than a single condition. What are your thoughts on this study? Do you think these subtypes capture the diversity of the autism spectrum? Share your insights in the comments below, ***.